As I have mentioned in the last class, the different softwares and tools used for bioinformatics analysis and the different databases utilized for carrying out the research work for different human diseases or required projects. So now I have decided to go for implementation of one of the selected software, that is ShinyGO, to implement how to utilize the database and go for further analysis for the purpose of carrying out your research project and finally develop into a research paper. And in the initial step, what you need to understand, how to utilize publicly available database and application of bioinformatics tools and the softwares for biological and clinical research as well as diagnostics. So here the title is Utilization of Publicly Available Database and Application of Bioinformatics Tools and the Softwares for Biological Clinical Research and Diagnostics Purpose. The selected databases could be Gene Expression Omnibus, GEO, cBioPortal or TCGA, GPR2, NCBI, SRA, EBI, Proteomics. GEO contains the data generated by microarray platforms, NGS or NGS platforms for different types of data sets. For example, gene expression, mutational data, whole genome sequencing, WGS, whole exome sequencing, WES, SNP profiling, long non-coding RNA, miRNAs, methylation data, ChIP-seq data, ATAC-seq data, or protein expression data, which could be extracted from EBI proteomics database. So once you decide what kind of data set you want to analyze, for example, here, we selected the differentially expressed genes list collection. So for this purpose, either you could go to gene expression omnibus if you want to go for differentially expressed genes, or if you want to go for mutational profiling, then select the mutational data set. And there is another portion, go to cBioPortal. Here I have selected the cBioPortal where the disease selected is AML, acute myeloid leukemia. And this outcome could look like here, the entire screen. So what we have done here, we sorted the mutated genes, which you could see here, 497 profiled samples. And all the top mutated genes, after downloading these top mutated genes, we processed for ShinyGO dataset. In ShinyGO dataset, this is left side is the top mutated genes. All these genes were pasted in ShinyGO web page and then processed for analysis, that is submit. After submitting, first outcome could look like this table where it is enriched pathways with the enrichment FDR scores, number of genes, for example, number of genes in each pathway or in the respective pathways, fold change or fold enrichment, and finally the name of that particular pathway. And these things are like chart, trees, network, egg, genes, plots, genome, string. All these things are multiple options for further analysis. So now in the next, so all these things are the output for the selected mutated genes list in case of acute myeloid leukemia. For example, here the output of ShinyGO for the selected genes of AML left side is the names of enriched pathways, and right side represents the tree for these pathways with the p-values followed. Here it is fold enrichment. Left side is each pathway has been plotted with the respective fold enrichment value. Now you can also plot the network of all the enriched pathways by using the same ShinyGO software. Now, if you want to analyze each individual egg pathway, for example, if you want to understand in this pathway at what are the genes which are altered or mutated in case of AML, and those altered or mutated genes are highlighted with red color, and the is chart is basically representing the location of all those genes in different regions. For example, cell membrane region, cytoplasmic region, nucleus region. So how you want to understand, and this is just for one pathway. You can also select all the pathways one by one. I have presented this figure just for better understanding and for clarity so that you can analyze all the individual pathways and the genes which are altered in each pathways and the location of the genes at different locations, for example, plasma membrane or cell surface, cytoplasm, or nucleus. For example, if you want to go for further analysis of all these mutated genes, you can go and click on gene details. Then you will get the gene names, different gene IDs, chromosomal locations, CHR, and the position in terms of mega base pairs, and the description of the genes, which is generally not required. And this is additional plots if somebody wants to plot the total number of like exons or transcript isoforms for each gene. They can also plot. It depends on the requirement. Now, this is the gene distribution, all the mutated genes and their distributions on different human chromosomes. And here you can see on chromosome Y, there is not even a single gene located, which means we can say in this particular case, the mutated genes do not belong to chromosome Y. And all other genes have certain number of mutated genes. For example, chromosome 19 has higher number of mutated genes with respect to the other chromosomes in terms of density. Now, if you want to analyze the further functional impact, like after the mutation in all these genes, what are the possible gene ontology terms which could be affected as a result of mutation? Then there are three different geoterms, for example, biological processes, molecular functions, cellular components, and this is the FDR values for each specific cellular components or biological processes or molecular functions, and then the number of genes which belong to a specific geoterms. Now, in the next part, you can also check the GET pathways and the number of genes, FDRs, and then go for further analysis of all the genes, like in terms of domain. For example, here it is interpro and then PFAM. Domain-based impact or analysis of all the genes or their inferred proteins could also be analyzed. So this is overall what you could do by using the ShinyGO software. Now, the next question will be that how could we generate the sufficient number of figures or outcome by using just one big software? And I think ShinyGO is one of the software or platform which could help you to develop one single manuscript for one specific kind of goals, where you can generate almost most of the figures from just this web server. So first thing, you generate the list of genes or proteins, then go for the processing of ShinyGO web servers, generate all the required outcomes which you want to highlight in case of your project work. For example, pathway enrichment analysis, the charts of all the pathways, trees, networks, kept pathways, genes, and the additional plots, genomes, string, and that figure could look like this. If you are not satisfied with the figures quality, you can also replot these figures by using some other softwares. For if you are familiar with coding, you can go for R plotting or MATLAB coding or Python or some other coding languages by using which you can plot these figures again in terms of better quality if you want. Or the most common thing is to plot the same figures by using Excel plot. And there you can also change the quality of the plots. But in these web servers, you can maximum generate in PNG formats. So this is overall what you could do by using ShinyGO. And this is very easy software for learning good bioinformatics to handle multiomics data and clear your hand and become a really good expert for analysis of multiomics data in terms of gene expression, mutation, or biological functions, networks, many more things. This is just one of the examples. And similarly, I will be presenting more softwares, outcomes, practical applications in the next classes. So this is overall what you could do.